I hope every one of you can see the screen. We are going to start with our class very soon, so kindly join faster. So, good morning, everyone, those who are in the class. I hope every one of you can see my screen. Can you all see the screen?
Yes, sir.
Yes, moreover, let me have the screen pointer. A laser pointer is better. This is the equipment with which we had discussed about the concept of agarose gel electrophoresis, right? I hope you all are now having a good idea about what is agarose gel electrophoresis. In this technique or in this equipment, we are trying to use a gel, right? And the gel that we have is usually made up of a carbohydrate called agarose. And that agarose was obtained, or we used to get this from algae like Gelidium and Gracilaria, right? Regarding which we already had the discussion. And agarose gel will be used in this equipment, and in a way there is use of electricity. That's why we have term electro, which is referring to the supply of electricity, right? Uh, and in a way, there is movement or migration of DNA occurs. And for that migration of DNA with the help of electricity, in that agarose gel is usually termed as agarose gel electrophoresis, right? And let's see whether your other friends are joining or not. Okay, so we have four. Okay, just I don't think, yes, let me check. Yes, now joining. Okay, now can we start with our class? Yes, I hope every one of you are here and able to hear my voice. Yes?
Yes, sir.
Okay, so I hope everything is working properly also. Let me uh, check here also. Who knows, this will be our reference, but still then the internet connection seems little poor. Now let's start. Before that, let me just check once, okay? Internet connection seems poor. That's why I can't proceed further. Before that, let me recharge and add on. Uh, Let me recharge quickly, okay? Uh, just a minute. Kindly give me 1 minute, okay? I'll be quickly recharging my data so that you won't face any problem of hearing. Okay, now I already recharged, and I hope every signal and everything is working properly.
Good
Yes.
morning, sir.
Good morning, good morning, good morning. So, shall we begin? Yes?
Yes, sir.
Okay. So, in the last class, we had a discussion about how we can purify DNA fragments, right? We are saying that we can isolate DNA from plant cell or animal cell or even from bacterial cell or from other microorganisms. So, after purification of the DNA or entire genetic material or genome of an organism from their cell, we are going to extract our gene of interest, right? And gene of interest will be DNA fragments. So how we can purify our gene of interest from the entire DNA of an organism, which is present in the nucleus of the cell of the organism. So for that, we are having an equipment which is shown below, regarding which we already had discussion, and with reference to the importance of the equipment and from the viewpoint of examination, we have to discuss once more so that your idea becomes more concrete. So here we are trying to discuss about agarose gel electrophoresis. Okay, agarose here is the carbohydrate. Any term ending with suffix ose is referring to carbohydrate. We have different types of carbohydrates. If I name few, glucose is ending with ose. We have fructose, sucrose, all these are OSE. So agarose is a carbohydrate which is obtained from algae, say like we have Gracilaria and Gelidium, which are used for extraction of agarose. And agarose usually is usually found in our labs in the form of powder. And if we mix it with water, and if we boil the water to mix it thoroughly, and if we try to cool it down, then it forms a gel. Okay, after preparing the casting gel, then we can extract DNA, okay, with the help of electricity. Okay, so how we can do that, regarding which we already looked at the equipment, different parts of the equipment. We said that we require electric supply, right? And usually our source or power supply is having voltage regulator. So this setup, this part which is attached with this tank, is your voltage source, and it is also having stabilizer. We can regulate how much voltage we want to supply. Okay, so this voltage source attached with a regulator is connected with two electrodes, and those two electrodes are usually having black cable and a red cable, and the black cable is representing our cathode, negative terminal, okay, and the red terminal or the red cable is usually denoting the positive terminal, that is anode. Okay, so you should remember that cathode is negative terminal, anode is positive terminal in case of this uh, equipment. Okay, now these two electrodes are usually joined together or connected together with the help of buffer. We are adding a buffer, a liquid, with the help of which there is movement of flow of electric charges. Okay, so, in addition to that, what you are seeing here is the gel tank. Gel tank is a tank in which we are also having a platform in between. There is a platform in between on which we are going to add our gel with the help of casting tray. So here the gel is prepared, in which there are tiny depressions, tiny pit-like structures, tiny holes, or these tiny holes are usually called as well. So the casting tray, as you can see here, it is placed onto the platform at the center of the gel tank, okay, in which we already added buffer. Buffer is a liquid which is containing some chemicals, and it helps in flow of electricity, it helps in flow of charges. And we are also having the agarose gel, and we can see tiny pores, tiny holes, and these tiny holes are the uh, holes, or these are the valves into which we are going to load, or into which we are going to add the DNA which we had extracted from plants, animals, or other organism. So after doing that, and if we supply the power, if we supply the electricity, then there is movement of the DNA which we had added according to the size of the DNA, according to the charge of the DNA, according to the mass of the DNA, from negative terminal to positive terminal. Why we have added DNA which we had isolated in the wells which are close to the negative terminal? Mainly because of DNA is a nucleic acid, and we all are knowing a concept where uh, all the acids are usually negatively charged, right? And bases are usually positive charged. So your nucleic acid, say like your DNA and RNA, so suppose here we are discussing about DNA, so DNA is negatively charged. Okay, I hope every one of you are following. Yes,
Yes, sir.
I hope every one of you can even understand what I'm trying to discuss here.
Yes, sir.
Okay, so what I had discussed so far is not there in your textbook, but these concepts are mentioned, and it's difficult for us to understand, right? So I had repeated in such a way that kindly consider, just to make you all consider, it's very important. Okay, particularly the movement of DNA, which is loaded at the negative terminal, and it is migrating from the negative terminal to the positive terminal on the basis of charge, on the basis of mass, on the basis of size. Those DNA fragments which are lighter, which are having smaller size, which are having less mass, are moving faster. And DNA which are larger, heavier in mass, these are moving very slowly. Okay, So this is the important concept. In addition to this, kindly remember DNA as negatively charged. Okay. So now let's try to continue with what we had studied regarding the agarose gel electrophoresis. It's important for us to see the DNA, right? Though we have seen the DNA, which is run here in the form of different colors, just to teach you the colors, we are seeing the DNA. Otherwise DNA is not colored, it's usually transparent. We may be facing difficulty in seeing it, so we have to find a solution in order to visualize, in order to observe the isolated fragments of DNA. So how we are going to do this in the molecular biology lab or in the biotechnological lab? We are going to do with the help of a dye which is called as ethidium bromide. So we are having dye. Usually dye is also known as stains in our lab, right? So we are having a carcinogenic, a cancer causing dye called ethidium bromide. So ethidium bromide is usually bright orange colored. Okay, and with the help of ethidium bromide we can see the DNA once we expose the DNA to UV radiations. After exposing ethidium bromide, which is added with the DNA, to UV radiation, then we can observe the DNA fragments. Otherwise, we won't be able to see it. So, dye usually, whenever we see it with naked eye, these are usually bluish in color. Okay, but once it is stained, even here the stain can be in different forms, right? Here it's little bright orange, right? So when we perform the experiment, when I was performing with the help of my friends in the lab in the colleges, we were seeing it as a blue colored stain, but here we are also having orange color, okay, according to the different products. Okay, but we usually say that according to your textbook you should remember it as bright orange colored dye. Okay, so once DNA fragments are separated, as you can see here, this is a cut portion of the gel which we had performed during the during our lab in college days. So this is a picture which I have in my laptop so that I could show here. Okay, so there is separation of DNA fragments, right? And you are seeing that these DNA fragments are showing us bright orange colored bands, and these bright orange colored bands are DNA fragments which are stained with ethidium bromide. Okay, so how we have seen this? By exposing the gel which is containing DNA added with ethidium bromide. And for visualizing this, we also require another equipment called as UV trans illuminator. So UV trans illuminator, as you can see here, this is UV trans illuminator, with the help of which we can see the fragments of the DNA. Okay, so here the most important part that you have to remember is about the dye. In order to visualize the DNA fragments, we have to use a dye or stain or coloring agent, and that is called as ethidium bromide. Okay, and for observing it, we require an equipment which is called as UV trans illuminator, regarding which you need not keep in your mind. Okay, so once we observe the stained DNA, we can see them as bright orange colored bands, mainly because of the color of the ethidium bromide. Okay, this should be kept in your mind. And just after doing this, in order to extract or isolate the gene of interest or the DNA fragment that we are required, we have to take it by cutting the DNA. Okay, suppose we require one portion, say like this tiny portion of the fragments of the DNA, we used to cut the gel and we used to extract, and we have to amplify it with the help of PCR technique. Okay, so here the process of cutting the gel and extracting or isolating the gel that we extracting the gene of interest or DNA fragment that we are required is known as elution. So we can say that elution is a process. As I always used to mention, any term ending with ION is a process. ING also is a process. SIS is also a process. So elution is a process as it is ending with ION. So in elution, what we are going to do is we are going to cut we are going to cut out uh, the gel part wherein our gene of interest or wherein our DNA which we required or desirable DNA fragment is present. Okay, so cutting off the gel part in which our gene of interest is called as elution. So kindly remember the term elution. You can even say that elution is a process wherein we are cutting out gel which is having gene of interest or which is having our desired DNA. Okay, and that desired DNA, after amplifying with the help of PCR, after making it into many of copies with the help of PCR, we can even use it for construction of our DNA. Okay, so the purified DNA fragments can be used for constructing recombinant DNA. How? By joining uh, gene of interest with the help of a vector, right? Suppose this is the gene of interest, then we can cut it and then we can join it with the vector DNA so that we can get a newly constructed recombinant DNA. And this can be used in order to introduce into host cell. And in a way, we can achieve experiment of interest. Okay, I hope every one of you are here.
Yes,
Yes,
sir.
yes, I hope every
Yes,
one of
yes,
you are
sir.
here and following all the instructions properly. Here, as I also already mentioned, the most important part is regarding the visualization of DNA fragment or gene of interest. Okay, for that we require a stain or a coloring agent or a dye called ethidium bromide. Though it is carcinogen causing cancer, it is useful for visualizing our DNA. Okay, and that is done with the help of UV light. Okay, and we also studied about elution as a process wherein we are cutting the gel in which our desirable DNA is located. Okay, can we proceed further?
Yes, sir.
Okay, so we'll proceed further. Okay, so let's have a short break in order to have the next topic. The short break here is mainly to ask you what exactly is transformation with reference to genetic engineering. Can any one of you answer what is transformation? It is a process, right? Ending with ION. Any one of you, can you answer? Yes? No volunteer? So I'm seeing Tenzi Desel here first.
Transferring the
Yes, yes, someone is trying to answer.
uh, transferring the genes of interest from DNA to RNA. Something.
Okay, transformation here is transform here is another term for change, right? Conversion. So transformation is earlier we already seen that there is transfer of newly constructed DNA, say like our DNA, into the host cell, right? So we can say that uptake of piece of DNA by bacterial cell or uptake of uh, DNA around the bacterial cell by bacteria is known as transformation. We already studied, right? So transformation is a process or it is a procedure through which there is transfer or there is introduction of piece of DNA. Okay, and that is introduced into host cell. So we can simply say that transformation is a process in which there is uptake of piece of DNA by bacterium cell with reference to the genetic engineering that we are focusing here. Okay, transformation is indirectly referring to uptake of piece of DNA or introduction of piece of DNA into host cell. Okay, how that is done, earlier we have already seen that first there is construction of our DNA. You can see here plasmid DNA with a gene of interest. All these are same. Okay, these are amplified constructed rDNA. And in order to introduce these rDNA into bacterial cell, what we are going to do is in the lab, we are going to take a Petri plate in which we have added the medium. We have added food for the microorganisms to grow, particularly bacteria, right? So bacteria are going to grow, and in these growing bacteria, we are going to add the newly constructed rDNA into it. by following all the aseptic condition, by following all the sterile condition, where we are maintaining every place in the lab clean, right? We are trying to maintain our ambience sterile, where there is no bacteria. So in the contamination free area, we are going to introduce these newly constructed rDNA into the growing bacteria. And as the bacteria multiplies, some of these bacteria take up the inserted DNA, inserted rDNA, and those bacteria which could take up the uh, introduced newly constructed rDNA, then those are called as transformants. Transformants means those who had changed, or we also call them as recombinants. This bacteria is now new because see, earlier they are having different structure, shape, right? The color is different, right? As I have represented. After the introduction of the newly constructed rDNA, these have changed. They have uptake. They have taken up piece of DNA, right? The piece of DNA here is mainly referring to the rDNA, and hence they have become transformant. Now they had changed, they had become recombinant because they had recombined the cellular characteristic, right? And in a way they are now showing different color, right? But some of these bacteria won't take up, won't undergo process of transformation, and these are usually called as non-transformants or non-recombinant. I hope you all are now clear about the term transformation. So transformation is changing of something, right? Or in a way, to be specific, we can say that there is uptake of piece of DNA, okay? Or there is introduction of a piece of DNA. Or we can even say that it is a procedure, it is a protocol, or it is a step through which we are trying to introduce a piece of DNA into the host organism. Here it is bacteria. As in genetic engineering, we are trying to use bacteria most of the time. Okay, so those bacteria which have taken up piece of DNA are called as transformants or recombinant. Those bacteria which haven't taken up the gene of interest, or those bacteria which haven't changed, as see the structure is same, right? The cellular characteristic is same like what it is represented here in the plate, petri plate. Though we can't see the bacteria like that in the petri plate, I've just represented for your understanding. Okay, so this bacteria which haven't changed is known as non-transformant or non-recombinant. So kindly try to remember this term as it is going to be used in the upcoming classes. Now we also have to understand transduction with reference to the transformation. Though not mentioned in your textbook, who knows? I've seen some questions related to it, as we CBSE usually follow CBSE textbook, that is NCRT textbook. Plus, we also have examiner. So in the examiner of NCRT, I've seen the term transduction. Okay, that's why I'm telling you. So transduction is a process in which we are trying to introduce gene with the help of virus. How it is done? First, let's look at the term. Transduction is transfer of genetic material from one bacteria to another, or from one cell to another, with the help of virus. So we have a viral vector. Virus will be carrying the gene, and it will be mediating, or it will be helping in transfer of genetic material from one bacteria to another. That can be also done in other living organism, not just bacteria. Okay, so as we are using bacteria most of the time in genetic engineering, we used to write bacteria here. Okay, so transduction is transfer of genetic material from one bacterial cell to another bacterial cell with the help of viral vector. Suppose this is the virus and it is carrying a gene of interest, and if we have to insert a particular desired gene into this bacterial cell, then virus will inject the DNA, and then in a way only the DNA which is required is injected into this bacteria. Okay, and that process is called as transduction. I'm saying that there is active role played by a virus, and that is viral vector. Why vector? Virus is carrying a gene. Now the gene which is carried by this vector will be inserted into bacterial cell. See, injection, and in a way this bacteria has now changed. Okay, this bacteria has now has had undergone process of transduction. So we can say that transduction is a process in which we have a virus which will be acting as vector of DNA, which is desired to be inserted into a bacterial cell, and we are inserting into the bacterial cell, and hence we achieve the process of transduction. I hope every one of you are here and following. Yes.
Yes, sir.
Yes, so we'll continue. Now, the important topic that we have to start and we have to discuss here is about your cloning vector, which is another important tool which is used in rDNA technology, right? And cloning here is a process of amplification, right? Process of copying the bacterial cell, or it is particularly referring to the copying of gene of interest. So cloning vector here is going to carry the gene of interest, right? So how we can say, what are, suppose say, like if you are told to enlist some of the cloning vectors which are used in rDNA technology, then we can say that the cloning vectors are plasmid, which we have seen right earlier, small circular DNA. We have bacteriophages. Bacteriophages are viruses that eats bacteria. That's why they are called as bacteriophages, right? And we also use cosmid. Cosmids are usually constructed, okay, constructed or synthesized vector, wherein we are trying to construct a vector which is hybrid of plasmid and lambda phage vector. A lambda phage. Lambda phage is a virus, and plasmid is a DNA, small circular DNA present in bacterial cell. So such hybrid or combination of plasmid and lambda phage is usually called as cosmids. So we can simply, what you have to understand here is examples of cloning vector which are used in rDNA technology are plasmid, bacteriophage, and cosmids. So the example here is showing you the plasmid vector, PBR322, which regarding which we'll be discussing later. Bacteriophages are also used as cloning vector, and then we have cosmids. Cosmids is having small circular DNA. Along with that, we also have DNA of lambda phage. Okay. Now, why we are using plasmids and bacteriophages as cloning vector? Mainly because of they are having ability to reproduce, or they are having ability to replicate, or they are having ability to copy themselves within the bacterial cell. In case of plasmids, these are native small circular DNA. These are present within the bacterial cell earlier, and they can multiply into many times. Okay, that's why we are using plasmid. Okay, and bacteriophages, as it is having also ability to multiply into thousands of times in a single bacteria without depending on other factors. Okay, that's why we are using. And if we stress more upon it, bacteriophages are used mainly because of they are having high number per cell, or we can say they are having ability to replicate uh, into large numbers in bacterial cell. Say like the other way of saying is they have high copy number. Copy number means number of copies, or number of them, number of the bacteriophages within the bacterial cell. See, what you are seeing here is a bacterial cell on which there is attachment of so many bacteriophages on its body surface, and then after infection, then there is entry of the genetic material of bacteriophage, and that is having high copy number, and it will be multiplying into thousands of the bacterial cell, and the bacterial cell will die. Bacteriophage will kill the bacterial cell, and many bacteriophages will come out. So that's why we are saying that bacteriophages can infect a bacterial cell, and they can multiply into thousands of times, and then they used to come out into number of copies. That's why they are having high number per cell. They are present in lots of themselves in one single cell, or we can say they are having high copy number. Okay, their number increases once they could infect a bacterial cell. Suppose if one bacteriophage infect or inject its genetic material into bacteria, then that single gene of the bacteriophage will be DNA of the bacteriophage will be integrating into the bacterial DNA or genome, and in a way that single DNA can produce thousands of bacteriophage, and they come out into large numbers. Okay, so we can say that uh, a bacterial cell may be having one or two copies of plasmid per cell, or some are having more than that, 15 to 100 copies. So that's why we are using plasmid and bacteriophages, as these are having high copy number. And high copy number of the cloning vectors will be enabling the scientists to multiply the gene of interest or rDNA into thousands of times. Okay, and in a way we can easily link the gene of interest with the vector, that is cloning vector like plasmid, so that we can have our DNA, right? So one of the major step of genetic engineering is construction of our DNA, which can be achieved by using a cloning vector called as plasmid, small circular DNA, along with the gene of interest, or we can use the bacteriophages. Okay, so we do have time, right? I'll just introduce you what is PBR322, regarding which a question may be there. So one of the synthetic cloning vector or plasmid is PBR322. So you can expect a question related to PBR322, and we can see here the statement itself is saying that PBR322 is one of the synthetic vector, and it is usually used a lot uh, in the rDNA technology. Okay, P stands for plasmid, as it is a plasmid vector, a small circular DNA which is obtained from E. coli. Okay, and BR stands for the name of the scientist who could construct or who could came up with this vector DNA. So we have BR. B stands for Bolivar, and one of the name of the scientist, Francisco Bolivar Zapata. And we have another R, R in the plasmid PBR322. R stands for Rodriguez. The full name is Raymond L. Rodriguez. So PBR322, P stands for plasmid, B Bolivar, R Rodriguez, and this is the number uh, uh, number of the name, the newly constructed plasmid name. Okay, number. So features of PBR322, we can say that it is having oricite. You can see here oricite, right? PBR322 is having oricite. Oricite here means origin of replication, which means this plasmid vector can multiply, can make it into number of copies with the help of oricite, where there is start of the replication of this small DNA. And we also have selectable markers. Selectable markers are the gene which are encoding antibiotics. We have AMPR, which is ampicillin resistant gene. This pink region of this DNA or small circular DNA is ampicillin resistant gene, which will be coding or which will be expressing a protein or an antibiotics called ampicillin. And we have other region of the pinkish region of the DNA, which is called as tetracycline resistant gene. This segment of the DNA, small circular DNA, will be making or expressing to form tetracycline antibiotics. So it will be making tetracycline resistant. Okay, so selectable markers that we are seeing here are genes which are coding resistant to antibiotics. Okay, and we are also having restriction sites or cloning sites. So we are having so many restriction sites here: PST1, PVU1, EcoR1, CLA-R1, HINT3, BAMH1, CEL1, and PVU2. So all these are restriction sites or cloning sites. Okay, and we are also having ROP. ROP here is referring to replication of plasmid, a replication of plasmid which is coding for a protein, and that is playing important role in the replication of this plasmid in addition to the oocyte where there is origin of replication. Okay, so these are the different parts, or these are the different segments of the gene. These are different genes or segment of the DNA which is constructed by Bolivar and Rodriguez. And the uh, synthetic plasmid vector that we have here is PBR322. Okay, so we have just 1 minute left, within which I would say that with reference to the uh, terms that we have studied here in this slide, we can say that uh, features which are required by a good cloning vector are it should be having origin of replication, it should be having oocyte, it should be having selectable marker, it should be having cloning site, and it should be able to clone into the host cell. Okay, so suppose if you are asked why is PBR322 a good genetically engineered cloning vector, we can say that it is achieving, or it is having all these different features of good cloning vector. Say like PBR322 is having so many features to make it into to achieve it as a good cloning vector, mainly because of it is having origin of replication, it is having selectable marker, it is having cloning site, and it is able to clone itself into host cell. So with this we'll stop here. In the upcoming classes we'll discuss about each of these different features which are required by a good cloning vector. So a good cloning vector example that we have studied is PBR322, which is constructed by Bolivar and Rodriguez. Okay, and this PBR322 is having oocyte, uh, replication of plasmid site, and there are many restriction recognition site, and we have two selectable markers, that is ampicillin resistant gene and tetracycline resistant gene, and these gene will be coding protein which will be making them resistant to those antibiotics, say like ampicillin and tetracycline. With this we'll stop here. Thank you so much. In case if you are having question, you can ask me or you can WhatsApp me so that I can help you. Okay, so we'll continue in the next class. I hope I can cover up this chapter. Even if I couldn't, we'll see. Okay, thank you all, have a good day.
Thank you, sir.
Thank you all.